Okay, so welcome back everyone to Stem Cells from the Sofa. This is our virtual seminar series that is designed to reconnect researchers and bring expert content directly to your sofa. Today's talk will be presented by Cristina Nostro. It's a great pleasure to have Cristina here for this episode of Stem Cells from the Sofa. Cristina joined the McEwen Stem Cell Institute as a principal investigator in September of 2012. She currently holds the Harry Rosen Chair in Diabetes Regenerative Medicine Research. Cristina's research program focuses on the differentiation of pluripotent stem cells to pancreatic cells with the ultimate goal of generating functional islet-producing beta cells for transplantation in patients with type 1 diabetes and the elimination of the need for daily insulin injections. Cristina has published numerous highly cited articles in journals including Cell, Stem Cell, and Nature Communications that have defined critical pathways leading to the efficient generation of human pluripotent stem cell derived pancreatic progenitors in vitro. And so on today's Stem Cell from the Sofa talk, Cristina will be covering a lot of background and with a talk entitled Modeling Human Pancreatic Development with Embryonic Stem Cells. Thank you, Cristina.
Thank you, John. Thank you for the kind introduction and for having me. Okay, guys, thank you all for joining in, and thank you for having me as the third speaker of this Stem Cell from the Sofa or from my living room. Today, I'd like to tell you a little bit about our effort on trying to understand human pancreatic development using human embryonic stem cells as a model. And the reason why we're doing this is that ideally, we also want to generate surrogate islets from embryonic stem cells or induce pluripotent stem cells for diabetes treatment. So, you know, diabetes is, let's see, okay. Diabetes is a global emergency. It's affecting more than 400 million people worldwide, and approximately 10% of this population is affected by type 1. The treatment for type 1 is insulin injection, but we know that pancreas and islet transplantation can actually work. This is a testament that cell therapy could potentially work as well for the treatment of diabetes. The problem with islet and pancreas transplantation is the donor shortage and the requirement for immunosuppression for the rest of the life of the patient. And so the future, the ideal treatment would be to generate surrogate islets from human embryonic stem cells or induced pluripotent stem cells and transplant this one day without the need of immunosuppression. And so human pluripotent stem cells, many of you know, have the capacity to give rise to all the cells in your body. And through probably 15 years, the last 15 years, we have learned how to use this embryonic stem cell in vitro to recapitulate these early stages of embryonic development. And so we can take human embryonic stem cells, differentiate them very efficiently to the definitive endoderm germ layer, which is the germ layer that will give rise to all the organs such as the lung, the stomach, the intestine, the pancreas, and the liver. And then we can pattern the definitive endoderm to what we know is the PDX1 positive endoderm. This population has the capacity to give rise to the pancreas, and we know that in vitro we can push these cells to become what we call the pancreatic progenitors. These are cells that have the capacity to give rise to beta-like cells in vitro and in vivo, and a population of cells that we define as polyhormonal that instead doesn't give rise to beta cells, but give rise to alpha cells. This is reminiscent of an early developmental stage in the mouse embryo that is known as first transition. The main difference between these two populations is the expression of one particular transcription factor known as NKX6.1. So you can see here by flow cytometry, the pancreatic progenitor population expresses both PDX, higher percentage of PDX positive cells express NKX6.1, while the polyhormonal cells almost lack expression of NKX6.1. In contrast, they express hormones such as C-peptide and glucagon. You see the polyhormonal cells here. And the pancreatic progenitor population in the differentiation system that we recreate in vitro do not give rise to any hormone positive cells. In the last many years, we've demonstrated together with other people that these polyhormonal cells actually give rise to alpha cells. So if we use a reporter cell line that has GFP under the insulin promoter, and this reporter line was generated by collaborators in Australia, Ed Stanley and Andrew Elefanti. So if you use this particular cell line and you sort for the insulin GFP positive cells and transplant the insulin GFP positive cells into immunocompromised mice, you realize that the potential of these cells is only to give rise to alpha cells. You see, they form this beautiful islet-like structure, but this islet-like structure only expresses glucagon and does not express insulin. This was shown by many people nowadays, and we know that this commitment is guided by the inhibition of TGF-beta signaling, both TGF-beta and BMP. On the contrary, the pancreatic progenitors has the capacity to give rise to beta cells, as well as all the other lineages of the pancreas. So if you take the pancreatic progenitors and you transplant them into immunocompromised mice, they will give rise to beautiful islet-like structures in vivo. You see insulin, glucagon, somatostatin, pancreatic polypeptide, but as well, they will give rise to ductal-like cells, as marked by the expression of cytokeratin 19, and trypsin, marking acinar cells. So what does this tell us is that we can actually generate pancreatic progenitor cells in vitro that have either a cell that has the potential to give rise to all the different lineages, or that pancreatic progenitors contain committed progenitors to each of the individual lineage. We don't know that yet. And just to put things into perspective, these pancreatic progenitors, or a similar population to this, is currently in clinical trial for type 1 diabetes, and a clinical trial run by the company Biosite. And ideally, I mean, the latest trial is using immunosuppression, but ideally in the future, as I said, we want to get away with this. And if we eliminate immunosuppression from this equation, we need to make sure that the differentiation of these cells and what we transplant into patients is actually a population that is pure and safe and clinically efficient to generate beta-like cells. We know that the efficiency of differentiation actually varies among different cell lines. In a study in 2015, we demonstrated that indeed, different cell line, if we measure the efficiency of differentiation by the formation of NKX6.1 positive cells, we see that some lines are particularly, we can efficiently generate pancreatic progenitors, while other line, like this particular IPS line, we cannot efficiently generate pancreatic progenitors. So we know in the field that different lines will behave differently, and this is due to their endogenous signaling. And so being able to understand how to control that will be key to producing a product that will be safe. Another alternative is, and this is something we were thinking of, what if we figure out a way to purify these pancreatic progenitors? Then it doesn't matter so much if we're not so efficient. And so the approach that we took, we thought we actually can potentially isolate these pancreatic progenitors if we identify specific markers expressed on this particular population. And our rationale was that to identify these specific markers, we had to compare the surface markers expressed in the pancreatic progenitors to a very similar population. In this case, we decided to compare it to the polyhormonal population. These cells are very similar. I told you that they both express PDX1. They're both pancreatic in nature, but only the pancreatic progenitors give rise to beta cells, while the polyhormonal does not. So to identify markers that are on the surface of this cell population, we teamed up with Dr. Thomas Kislinger at the Princess Margaret Cancer Research Center and MVP Molecular Biophysics in the Department in Toronto, to analyze the surface marker by mass spectrometry analysis. We actually provided Thomas with 3 different populations: the pancreatic progenitors, the polyhormonal cells, as well as undifferentiated human ES cells. And so the proteins were digested from this different population, were digested into small peptides. And we decided to enrich these peptides for the glycosylated peptide using a magnetic hydrazide bead. The reason for enriching for the glycopeptides was that all the proteins that are found on cell surface or surface on membranes are glycosylated. So by enriching for glycosylated peptides, we would enrich for proteins that we would find potentially on the surface of the cells. So the glycosylated peptide were enriched by magnetic separation, and then the magnetic beads was released by deglycosylation, and the peptides were run through a mass spectrometry analysis. And I have to say this analysis was done by a very talented PhD student, now as a postdoc in Germany, a PhD student in Thomas' lab. His name is Ankit Sinha. And the result from this analysis was, you know, if anything, probably better than what we expected. The unsupervised hierarchical clustering separated the undifferentiated ES cells, as you can see, running triplicate, away from the pancreatic lineages, the pancreatic progenitors, and the polyhormonal cells. But most importantly, it identified clusters of peptides that were specifically expressed in one of these different subpopulation. So here you see cluster 1 are peptides that are expressed in the undifferentiated ES cells. Cluster 2, peptides expressed in the pancreatic progenitor population, and cluster 3, peptides expressed in the polyhormonal cells, and so forth. And of course, we decided to focus on the peptides that were found to be expressed in the pancreatic progenitor population. And we mined this data with the idea to look for proteins for which we could find antibody that could be used by flow cytometry. And one of these protein was GP2. We validated the expression of GP2 and demonstrated that indeed GP2 is expressed in the pancreatic progenitor population and not found, or virtually not found, in undifferentiated ES cells and polyhormonal cells, suggesting indeed that it is a specific marker for this pancreatic progenitor population. We had no idea at the time what GP2 was, so we searched the human atlas, and we found that GP2 is expressed, is a marker of acinar tissue, is expressed in the adult pancreas only in acinar cells. You see here islet of Langerhans that are negative for GP2. Which of course prompted the question, right? We found GP2 to be expressed during human ES differentiation in a particular population that is only found in development, because the adult pancreas lacks the presence of these pancreatic progenitor cells. So the next question was, is this GP2 really a marker of a pancreatic progenitor, or is just an artifact of cell culture? So to address this, we actually teamed up with a group in Vanderbilt, Al Power, Marcela Brisova, and Diane Saunders. And we asked them to stain human pancreas, postnatal human pancreas, for GP2, to see if indeed GP2 was marking and tracking with the expression of NKX611 in a population that could represent a progenitor cell. And so this is a beautiful staining of a gestational week 33 postnatal pancreas, where you see in blue insulin producing cells. These are the nascent beta cells. They are expressing insulin, and NKX6.1 in red. They're at the center of this developing pancreas, section of the pancreas, and surrounded by GP2 positive in green cells. Interestingly, I hope this projects well in your computer, but you will see that there are GP2 positive cells, like I'm showing you here. They are co-expressing NKX6.1. And those would be the equivalent of the cells that we generate in vitro in the differentiation. And interestingly, you also see GP2 positive. They're negative for NKX6.1. And we think these are the cells committed to the acinar lineage, while you also can, if you can appreciate, some NKX611 that don't express GP2, they don't express the insulin, but they're probably committed to the endocrine lineage. If these cells is really putative multipotent pancreatic progenitors, the cells should also co-express another marker, and that would be PTF1A. So we know that in the mouse pancreas, co-expression of PTF1A and NKX611 tracks with the multipotent pancreatic progenitors. Whether GP2 and the existence of these cells is replicated in the human pancreas, we didn't know. And in this staining performed by Diane and Marcela Bersova, we see cells indeed co-staining PTF1A and NKX611 and co-expressing GP2. And I think this image is particularly powerful for me because it's suggestive that indeed GP2 could identify the putative multipotent pancreatic progenitors in humans. The other very important message from this picture is that contrary to what we know from the mouse, where these multipotent pancreatic progenitors only exist for a very, very short time in development, in human, maybe the multipotent pancreatic progenitors persist for a very long time. So it arises very early in development, and it may stay there until birth. So to go back to our differentiation culture, we wanted to check the kinetic of expression of GP2 throughout stage 4, which is the stage where the pancreatic progenitors are generated. And to do this, we use another reporter line that has GFP under the NKX611 promoter. This was also generated by our collaborator in Australia. And so you see that GP2 starts to be expressed before NKX611. You see at day 10 there is a considerable population of GP2 positive cells that is negative for NKX611 GFP, and as soon as NKX611 GFP starts to be expressed, these cells are positive for GP2. This is not something that we see only in the reported line. If we stain an untargeted line, H1, you'll see that we find GP2 positive and NKX611 positive cells also co-expressing PDX1. So this is true for various lines, and this marker could really identify the pancreatic progenitors in vitro. So can we use it to enrich for these pancreatic progenitors in the differentiating culture? To do this, we actually sorted for GP2 positive and to compare them with the GP2 negative and unsorted population, the pre-sort. And we further differentiated the sorted population using protocols generated by Rezani and Pagliuca in 2014 to generate beta-like cells in vitro. The results was very satisfying because indeed the fraction, the GP2 positive cells, generated higher percentage of beta-like cells, which are characteristic by the co-expression of C-peptide and NKX611, higher frequency compared to the unsorted population, the pre-sort, and the GP2 negative population. Interestingly, for those in the field, I think the GP2 negative that give rise to the beta-like cells are those cells that maintain NKX611 expression but commit lose and GP2, and so they're a little bit more advanced compared to a GP2 positive NKX611 positive population. These cells are monohormonal. They have all the characteristic of the beta-like cells. And the next question we had was, if we sort for GP2, can they also give rise to beta-like cells in vivo? And indeed, they do. Here are the GP2 enriched populations sorted by magnetic separation. And we show that they give rise to C-peptide positive, glucagon, and somatostatin positive cells. So they give rise to all of the endocrine lineages, but also they maintain the potential to give rise to ductal cells as shown by cytokeratin 19, as well as acinar tissue. So even in the enriched GP2 population, we still have progenitors of all the different lineages of the pancreas. just to summarize this part, I want to just take a moment to pause and you know, go over what I showed you. And first of all, human embryonic stem cell differentiation provides a powerful platform to study human development in vitro. So the identification of GP2 as a marker of the putative multipotent pancreatic progenitors would have not been possible otherwise. We demonstrate that this marker can allow for the enrichment of a population that has the potential to give rise to beta-like cells in vitro, and the GP2 enriched population also maintain their developmental potential and give rise to all the lineages of the pancreas in vivo. But what I told you in the beginning is that, you know, we are differentiating these human embryonic stem cells to different lineages. We want to generate safer population for the clinic one day, but also we want to understand how lineage commitment occurs. And so, you know, early on, we demonstrated that it's through TGF-beta inhibition that we can actually push the formation of these polyhormonal cells and eventually give rise to an alpha cell phenotype in vivo. And recently, we demonstrated that it's through Wnt inhibition that PDX1 positive endoderm commits to the pancreatic progenitor cells. These are studies by Frankie Poon, a PhD student in the lab, who showed that if you treat the PDX1 positive epithelium like cells with a strong Wnt agonist, in this case we were using Kyer, you block the formation of the PDX1 positive and KX611 positive cells. In this graph, you see the stages throughout the days of differentiation during stage 4. And you don't see the line here for the Kyer treatment, but there is no expression of PDX1 and KX611. In the presence of a Wnt inhibitor, WIKI4, the cells commit to the PDX1 positive and KX611 positive cells and give rise to a very high frequency of PDX1 positive and KX611 positive population, which will then be able to differentiate to beta-like cells in vitro using the protocols developed by Resani and Pagliuca. And here you see the commitment to beta-like cells is the C-peptide positive and KX611 positive cells are only generated in the WIKI4 treatment. How does this happen in vivo? You know, we have no idea. And for this, we have to thank the amazing collaboration with the lab of Tai Kim and a wonderful student, Theodora Young, who elucidated how this tight control of signaling is maintained in vivo. And so what they was what Theodora and Tahi were able to demonstrate is that the pancreatic mesenchyme, contrary to the intestinal mesenchyme and the stomach mesenchyme, is able to maintain very low level of Hedgehog signaling and Wnt signaling throughout development of the pancreas. And this is key for the proper commitment of the pancreatic epithelium. So if they specifically knock out two important regulator of Hedgehog signaling, SUFU and SPOP, in the mesenchyme, as you can see here, they actually lost commitment to, or they reduced the commitment to beta-like cells, as you can see here. Importantly, by losing SUFU and SPOP in the mesenchyme, they upregulated Hedgehog signaling. You see here downstream effector GLI1 and PATCH1, and importantly, increased Wnt signaling molecules. And this increase in Wnt signaling was inhibitory to the pancreatic development. So here is the schematic of how we think this is happening. The pancreatic epithelium is controlled by the stromal, the pancreatic mesenchyme, by low level of Hedgehog signaling and low level of Wnt signaling. If this mesenchyme loses control of Hedgehog by losing SUFU and SPOP, Wnt signaling are activated and they block the growth of the pancreatic epithelium at the stage of PDX1 positive and KX611 positive PDX1 positive cells, and there is the impairment in beta cell commitment. With this, I summarize this other part, really telling you that human embryonic stem cell as differentiation is allowing us to elucidate key signaling molecules that are important for pancreatic development in humans, And the results of the work that we generated in vitro can also be verified with beautiful and elegant developmental studies in the mouse. And with this, we demonstrated that Wnt inhibition is particularly crucial for the endocrine pancreatic progenitors generation and endocrine commitment. And I'll finish with the slide that is the most important. And here is my group, a wonderful group of talented students and postdoc and technicians. This is when we were all together at work. Hopefully we'll be back together soon. Big thank to all the fundings and collaborators, and I'm happy to take questions.
Fantastic. Thank you very much, Cristina. Great talk. So we've got some questions coming in. Francis Lynn is asking, Heiko Lickert's group recently showed that pancreatic progenitors could be distinguished at the definitive ender stage with CD177, and others have shown that sorting of beta cells is possible. What stage do you think would be best for sorting enrichment of a cell therapy?
Hi, Francis. Thanks for your question. You know, that was our original thought, that sorting would be the solution for a safe and a clinical product. The reality of the sorting as we are dealing with it at the moment is that sorting would probably end up with us losing too many cells. It might not be a viable way. What these markers can actually do for us is to help us guide the differentiation and make sure we're on the right track, almost like quality control steps. If one could sort, you know, you would sort at the time where you have the highest number of cells. And so whether this is at the definitive endoderm stage, where you have like, you know, a higher number of cells might be easier. Pancreatic progenitors is also like, you know, increased number. As for the more you progress, the more, you know, at least in our hands, you lose cells. And so the efficiency might be more, not as good. I hope that answered your question.
Do we have more questions coming in? Please do use the Q&A button at the bottom to type in your questions and ask Cristina. It's a great opportunity to pick her mind on this. So Cristina, tell me about the maturation status of the cells that you're able to derive using the technique that you've optimized here. So
what we published so far were cells that were not actually functional, and you know, through a series of papers, you know, from the Millman groups, for example, we've now learned, and Melton, we've now learned that dissociating and reaggregating the cells towards the end stage of the differentiation, actually, and culturing them in the absence of certain factors, actually allows them to reach a glucose responsive state. I think everyone in the field would agree that we still don't have a fully mature bona fide beta cells. There are certain factors that are still lacking, and so I think we're all striving to generate something that is, you know, more closely resemble adult beta cells. Whether that is necessary for treatment purpose, I would argue maybe not, because in reality, we all know that once we transplant the cells, they are able to, you know, start functioning relatively quickly in vivo. So there is something in the in vivo environment that will actually push them towards maturation. So, you know, if you have, you know, maybe a not so committed cells, but you have more of them, it might be a better way to pursue a clinical product. It's a balance, I think, between cost efficiency of differentiation, if you want, and generating a good product, if you know what I mean.
Okay, so we have another question come in from Christos Karampaleis. Sorry if I butchered your name there. Great talk. The expression of GP2 cells, positive cells in the human stainings look to be stronger on one side of the cell in certain epithelial structures. Do you think GPT2 could be involved in the polarization of epithelial pancreatic progenitors and affect differentiation in humans? Greetings from Sweden.
Chris, that's a very good point. I actually, despite having looked at those images for so long and being amazed every time, I never thought about that. It's certainly something that, it's interesting. We've never, I don't know whether GP2 has been linked to, has been demonstrated to be playing a role in polarization, but it's something interesting. I'll look into it and maybe we can chat about it.
Okay, so we have one more. PROCR positive pancreatic progenitors from adult mice were recently demonstrated in, published in Cell 2020 to generate islet organoids in vitro. Does this marker align with the GP2 during pancreatic progenitor generation from embryonic stem cells?
Unfortunately, we don't know. I mean, it's after that paper came out, I think everyone in the field, this data was generated in mouse, and it was never tested whether PROCR is expressed in human islets. It's something that I think is in the mind of many, many people, and we haven't looked in whether this is even expressed in human cells. But I think this is something that many people will look into.
Okay, thank Cristina very much for her talk. It was excellent. Thank you, Cristina.
Thank you so much. Thank you, John. Thanks for having me.
And I'm just going to give you a heads up that our next Stem Cells from the Sofa event will be held at 1 p.m. on May 13th. That's in a week's time, not the usual 2 weeks' time. And will feature Dr. Jim Woodgett, who's the Director of Research and a Senior Investigator at the Lonnenfeld-Tannenbaum Research Institute, part of Sinai Health System. Jim is part of a working group that has been tasked by the Toronto Academic Health Service Network to formulate a plan for returning to normal research activities following COVID-19 shutdown. Jim will be sharing his insights regarding the reopening of research labs in the next Stem Cells from the Sofa. Okay, thank you very much.